Hello, everybody. We still got 2 minutes and waiting for some more committee members to join. Julia, I presume you can hear me now. I think everyone was on mute earlier. Hello, Virginia. Thank you for joining. Looking for Jim in the list. And then I think we will have all our committee members. Zach, FYI, after your seminar, we'll have to figure out, maybe Teresa, if you're hearing, we'll have to figure out how to get our committee members as panelists. Apparently the link that we sent out was actually just for attendees. That happened to me as well, even though I was obviously a host. So we'll have to figure that out afterwards.
Okay, I can promote people who need, yeah, and I think I can turn over hosting to you. If worst comes to worst, I'll just create a new meeting for you guys to take. Yeah.
Okay. I could send you the list of people.
Perfect.
I'll do it by email now.
Thanks.
So it is 9 o'clock. Hello, everybody. We're still waiting for one more committee member. I'm browsing through the list of attendees, which is growing rapidly. There. Hello, Jim. I think we have everybody, and therefore I think we are set to go. So I think to give Zach the full hour, I'm not even going to wait as more and more people stream in. Hello, everybody, and thank you for coming. This is a special day for us in the Novak Lab, and it's a bittersweet one, a special and bittersweet one for me personally, for many reasons, but simply it's been an absolute joy to have Zach in the lab, to work with him and to learn with him. And this marks the end of a special period for us. Looking back, for one, to me, it's simply inspiring to see how efficiently Zach has achieved all of the goals that he set out for himself when he joined the lab. Not in terms of the research per se, but in the terms of the things he wanted to learn big picture. Now, Zach was already an accomplished ecologist, field biologist, kelp forest ecologist when he joined. I think he had something like 300 scientific dives under his belt. He was certified in rebreather technology, trapping sea otters by sneaking up from them below with Navy issued rebreathers on his back. And I could go on with all the things that he'd already had in his CV when he joined. But what Zach wanted to do when he started his PhD was to learn quantitative skills and to get a better understanding of theory. And as you'll see shortly, he accomplished those goals in spades, not only in terms of a novel use of time series, in terms of theoretical modeling, but even adding Bayesian statistics in a somewhat non-standard way into the mix, non-trivial ways of using these tools. And he accomplished all that while at the same time working on managing projects totally unrelated to his dissertation, things that he had brought in with from his undergrad and tech days. For example, working on developing flipper tag technology for sea otters to track infer their social networks and interactions and behaviors developing these cutting edge sensors and tags to put on the tags on the flippers of sea otters. But today's talk is not about sea otters, but about work that I expect and hope will be listened to by kelp forest managers in particular. Community ecologists in general, I think, will pay attention, but kelp forest managers need to be listening to things that Zach has done and the things that his work is showing. This is important and timely work, given all that is happening in kelp forests, not just along our coast here, but around the world. And I think it offers a new perspective and a much needed additional perspective on how we should think about how these systems function, and therefore how to manage and conserve kelp forests around the world. I think I'll leave it at that. Give you as much time as possible, Zach. Take it away. Yeah, good luck. Enjoy.
Okay. Well, thank you, Mark. I really appreciate the kind words. It's very nice of you to say. And likewise, it has been an absolute pleasure learning and growing with you in your lab. So thank you. All right, let me start a timer. And can you all see a laser pointer? I think you can. Okay. Well, hello, everyone. Thank you for joining us today. My name is Zachary Randell, and today we're going to talk about how grazer resource behavior, interactions, and feedbacks control kelp forest dynamics and stability. So first, for chapter 1, I'm going to talk about some motivation. I'm going to lay out some ecological concepts, specifically species interactions, disturbance, feedbacks, and alternative states that are themes throughout my dissertation. And then I'll introduce kelp forests, my study system. Chapters 2 through 4 will talk about the main research, kelp forest dynamics controlled by substrate complexity, urchin behavior structures kelp forest stability, and drift algae controls urchin behavior. So we have a roadmap up here at the top. And if you have questions, comments, concerns on topics with specific slides, just note the slide number and I'll pop right back to it in the question session. All right. So we as ecologists care about the structure and functioning of ecological systems, with structure being what species are present in their abundance, and where functioning is the interactions and processes connecting these species. We especially care about how and why community structure and function change through time. Species interactions are fundamental towards maintaining community structure. This topic has received extensive attention, and we now understand that species interactions are not fixed, but rather are dynamic, responding to a host of biological and physical factors. For example, it's been shown that the hunting spider indirectly increases plant survival by its presence alone, even when its feeding structures are glued shut, such that it can't consume anything. And this is because grasshoppers avoided the spider. They changed their behavior and thus did not consume the plants. Other research has shown how habitat complexity, so the structural complexity of rocky reef, can also modify species interactions. It's been shown how sheephead wrasse aggregate around rocky reef, focusing predation pressure on urchins and other invertebrate prey in areas of high complexity. I also want to motivate disturbance, which shapes community structure, and we can gain insight from studying how communities and ecosystems respond to perturbation events, so the removal of individuals from a community or ecosystem. Now, lastly, I want to motivate stabilizing feedback processes, as they are crucial towards maintaining a resilient community structure. For example, seagrass growth traps particulates and nutrients, thereby making the water less turbid. This increases light availability, which further supports additional growth. So seagrass thus provides stabilizing feedback to itself. Now let's bring these pieces together. When a system is perturbed, the eventual recovery of the original state is dependent upon the integrity of the feedback processes stabilizing that system. If those processes are inhibited or suppressed, different feedbacks can strengthen, and in doing so can stabilize a new arrangement of community structure. We sometimes call these alternative stable states. So my dissertation at a high level examined how species interactions are affected by and themselves comprise stabilizing feedback processes, and how disturbance can upend those feedbacks to elicit switching between alternative stable states. Now, I pursued these concepts in temperate, cold water kelp forests, which are incredibly diverse and productive and support numerous species of recreational and commercial interest, as well as those of conservation concern. Urchins are a key herbivore that consume, among other things, kelp, such as the giant kelp species beautifully depicted here by Embroidology SB. Just a quick shout out to her for allowing me to use her beautiful design all throughout this presentation. Urchin behavior, however, is not fixed. Instead, urchins exhibit a behavioral switch. They often remain inactive, so they're not out wandering around, they're not grazing kelp, and instead they tuck away into cracks and crevices, often consuming drift algae. I'm going to symbolize drift here, and drift is simply detached pieces of kelp or other aquatic plants that for one reason or another is no longer attached to the seafloor. Many species, such as urchins, consume drift for food. So urchins are inactive at times, then something happens or a combination of some things happen. They leave shelter and actively move across the substrate, grazing kelp, and this can lead to the formation of what's known as an urchin barren. Now, crucially, in exhibiting this behavioral switch, urchins alter the strength of feedback processes being expressed. And that's because urchins graze kelp, thereby suppressing the future potential for kelp to recover. Now, we know that when urchin predators are present, they exert direct and behavioral effects upon urchins, thereby limiting urchin consumption of kelp. And when those predators are removed or significantly depleted, urchins can manifest their behavioral switch. But there are also instances in which urchins switch behavior and form urchin barrens with no underlying change in the predator population. So it seems we're missing a piece here. As an example, let's examine a 38-year time series of giant kelp. And we see these beautiful kelp population cycles, and we're able to formally evaluate that cyclic behavior down here. And we have reason to believe that these cycles are from kelp competing with itself, essentially, and I'll talk more about that later. The point is, this time series is from a very healthy kelp population where urchins are present, but they're not forming a barren. In contrast, in just a couple kilometers away, we can view another time series where kelp is present, but there's also this period where kelp has been wiped out for over a decade. And as I'll go on to show later, this is switching back and forth between the forested, urchin barren, and forested state once more. What I want to emphasize here is that these different dynamics manifested around the same island, a very small island subject to the same potential predator exposure, and likewise the same broad set of environmental conditions, yet we see fundamentally distinct dynamics. So again, at a fairly high level, my dissertation evaluated these distinct dynamics and explored how the availability of drift algae, along with the processes of drift production, retention, and loss, affect urchin behavior, and thereby the emergence of alternative stable states. Now, you may be thinking, this is all well and good, why do we care? We are seeing firsthand the consequences of an increased frequency and intensity of disturbance events often associated with climate change. We saw the spike in warm water, the anomalous blob event along the Northeast Pacific coast. Sea stars melted in a marine disease emergency. Kelp was wiped out. Urchins rose to dominance, and abalone starved to death, resulting in the closure of multiple fisheries. In response, numerous conservation and restoration efforts marshaled to try to reverse or stave off this switch to the urchin barren state. So it really is important to try to understand not only the processes that elicit these switches in state, but those that also stabilize the respective states, i.e. the feedbacks that stabilize both the forested and the urchin barren state. So with that, my three primary chapters I'm going to talk about. Chapter 2, kelp forest dynamics controlled by substrate complexity. I evaluated how do kelp forest dynamics vary across substrate, across space and time, and how does substrate complexity associate with those distinct dynamics. And then I hypothesized how substrate complexity is modifying feedback processes, and those hypotheses are what I then explore in the latter two chapters. Specifically, I model the interactions between kelp, between urchins consuming kelp in drift, and test how a preference to consume drift affects the system dynamics that emerge. And I also evaluate how drift production, retention, and loss modifies kelp forest dynamics in terms of a persistent forested state versus switching back and forth between alternative states. And finally, with my last chapter, I tested a key assumption from chapter 3. Do urchins indeed prefer to consume drift? And does the density of drift control the consumption of kelp? I also evaluated whether urchins exhibit a switch between kelp and drift. And I evaluated this with a subtidal cage experiment underwater using scuba. All right, first off, kelp forest dynamics controlled by substrate complexity. So I analyzed a time series of subtidal community data from San Nicolas Island and the Channel Islands off the Southern California coast. Shout out to Mike Kenner with USGS for maintaining this ongoing program for decades. It is an amazing time series, and this is an amazing group of people. It has been a privilege to work with them over the past 10 years. So I analyzed data from 6 subtidal stations around this island, and there's a couple different types of data collected, but today we're just going to talk about these 10 by 2 meter swath transects, where we counted the abundance of several sea star, grazer, and kelp species. And what I want to emphasize is that these transects are permanent, so there are bolts in the seafloor that we go back to. So we're counting these species twice a year, year after year, and we're able to see how these 10 by 2 meter slices of seafloor change over the decades. Now, working out there, one thing that struck me was just how distinct these sites were, even within a single site. There are areas where the substrate is completely flat, there are boulder piles, and there are dramatic pinnacle structures. And it appeared that there were distinct patterns of community structure across these different types of reef. So in an attempt to capture some of that substrate complexity variation, I used a magnetic surveyor's wheel. So there's a little wheel and a magnet inside it, and a sensor detects the magnet and records how many rotations the wheel makes. And so I quantified the substrate complexity, or the linear relief or rugosity, of all these 10 by 2 meter transects. So here we see a 2 meter transect, a straight 10 meters as the crow flies or as the fish swims. And I simply rolled the wheel along the seafloor and quantified how much linear relief or rugosity is contained within these straight 10 by 2 meter transects. Now, to analyze community structure, I use non-metric multidimensional scaling. Don't worry about the details of the analysis. Just know that for all the dots you see and are about to see, each dot is the community structure of a single transect at a single point in time. 2 dots that are very close together exhibited a very similar community structure. 2 dots that are far apart exhibited a very different community structure. Now, let's look at some dots. Do not worry about color. Color are the different sites. What I want you to look at is the size of the dots, which is the abundance of giant kelp at each one of these transects. And as we can see, there's lots of kelp here, and on the other end of this ordination, very little kelp. Let's look at the same thing, but for the abundance of purple urchins. We see there's barely any urchins here, and there's a lot of urchins over here. And in the middle, we have this mixture where kelp and urchins are both present, but urchins here aren't forming a barren. And indeed, when you dig into these data, this area over here, you can even see it's just slightly separated. This region of ordination space are transects exhibiting the urchin barren state. And on the other hand, on the other side, we have transects exhibiting an algae only. So pretty much it's kelp and no urchins. And then again, we have this broad mixture where kelp and urchins are both present, but urchins are not forming an urchin barren. Now, this appeared to me to capture a gradient in community structure from forested mixture to an urchin barren state. And because of that, I can make the claim that this captures the state of the system, the system state. And in doing so, that opens up some additional options for us. This is because we can think about system state as a stability landscape, with deeper basins corresponding to more resilient arrangements of community structure, and shallow basins corresponding to less stable states. Now, the landscape can shift as environmental conditions change, or when environmental conditions are constant, a system can be perturbed. It can experience a disturbance event. Say we can remove a bunch of individuals, or a bunch of kelp is removed from a large wave event, and that jolts the system into a different state. Here's the key part. Because systems are constantly perturbed, so you can think about the ball vibrating, the species are slightly changing, their abundances are changing and shifting due to demographic and environmental stochasticity and occasional large perturbations. Repeated measurements of system state across space, time, or both can reconstruct the stability landscape of the system. So here, if this is a frequency distribution of system state, so repeated observations of system state, then its inverse is the stability landscape of the system. So this approach is called potential analysis, and this is what I was able to do with our two-dimensional ordination. Specifically, because axis 1 captured this system state, this community gradient, I simply collapsed this two-dimensional ordination down to a single x-axis and then visualized the resulting kernel densities. And what we see here are representations of the stability landscapes for not only our individual sites, but the transects that comprise them. So you see 5 different colors. Those are the 5 different transects comprising each of the 6 sites. And so one thing that'll jump out here is that we see some sites exhibit multiple modes, or there's multiple states exhibited. Other sites only exhibit a single state. And so just to orientate you, these are the same as what was in the ordination, in the sense that this middle area is the mixed kelp urchin state. The far right is an urchin barren state. And 2 sites exhibit the third, this algae-only state, which is rather bizarre, and I'll briefly touch on that. Although this other site also exhibits that same state, and only that state, and no shifts between. So this transect here is doing something completely different than all the other transects at the same site. And it's unique enough that this little guy got its own section in a write-up. So all right, so we have these different states. Now a step further, because we have temporal information, I also calculated the velocity of community shift through two-dimensional ordination space. So that's just taking the distance traveled through as the community changes, and then divide by the time elapsed between the sampling points, and then we have a velocity or rate. And now the lines here, the lines are the average velocity given the placement of the starting and end points of the time. So this is the average velocity across the stability landscape. And what I want you to see is that within a basin of attraction, the velocity is low. So this is akin to the species abundance is just kind of changing a little bit. This is the ball vibrating slightly. But the system and an attractor is dominated by stabilizing feedback processes. So it's not going to want to change. It's going to take an intense disturbance event or a heavy perturbation. So you can think about an attractor as having a gravitational pull, and it's going to take quite a bit to perturb the system into an alternative stable state, an alternative attractor. That's at least what we would theoretically expect, and that is indeed precisely what we see, which was a very exciting finding to recover. Do not be deceived by our friend here. This looks like high velocity switching. This is just one transect doing something completely different. There is no switching in state for this site. And then one of my favorite points, this site and this transect, one of the transects exhibited a high velocity movement away, so out of the basin of attraction, and then the next time period was right back to where it started. So it was perturbed and then nearly instantly, in terms of our observations at the 6-month temporal scale, recovered. That is a definition of resilience. Okay, What about substrate complexity? So now these are those rugosity values that I measured. So 1 here on the y-axis, that's a 10-meter transect that truly was 10 meters when I measured it, so it's completely flat. On the other end, we have a 25-meter transect, or there's 25 meters of substrate complexity within these 10-meter transects. So this is massive pinnacle structures up and down, just beautiful rocky reef. And I've ordered these data and all the data by substrate complexity. So we see it is only the low rugosity sites and transects that exhibit these shifts in state. And similarly perturbed high complexity locations do not exhibit shifts in state through time, or when they do shift, they rapidly recover. Now I'm just going to quickly show you essentially the exact same data as what's represented in the top panel. I'm just going to expand down the movement across that state axis. So now we're looking at the temporal dynamics. And I'm not going to spend too much time here, but I want to motivate that we think we have evidence for transient dynamics. That is the gradual, continuous recovery shifting away from that algae-only state back to a mixed state as urchins eventually recover following a massive, massive disease event. And I also want to quickly motivate that we think that urchin density is not driving, or at least not the sole driver, affecting these shifts in state. So we're now visualizing total urchin abundance on the y-axis within each state. So M, the mixed state, B, the barren state. And what we see here is there's a huge amount of overlap. There's plenty of sampling instances or sample points in which there's a ton of urchins in the mixed state, but they're not forming urchin barrens. All right, and I lastly just want to motivate, in case you are thinking, what else could be driving these patterns besides substrate complexity? I evaluated a variety of environmental variables as well as other species, and none of these variables associate or covary with both either substrate complexity or the kelp forest dynamics that we observed. So we do think there is something going on with substrate complexity and the associated stability patterns that we're seeing. Okay, so what do we think is happening here? I'm not going to motivate all of this slide. I'll come back to it at the end. But in short, these low complexity locations exhibiting alternative stable states, we essentially think that the processes of drift production and retention are prone to getting perturbed. So a large wave event comes through and just blasts these sites clean. We've seen it. The kelp is removed, the drift is removed. With no food, urchins exhibit their behavioral shift. They come out, they graze, they form the feedbacks that stabilize the barren state, and voila, you're in an alternative stable state for a decade potentially until disease comes through and wipes them out and kelp is able to recover. Similarly perturbed high complexity locations do not exhibit this switch. Now we think there's multiple feedbacks that are being stabilized by substrate complexity. One of them is the retention of drift algae. So the drift, it gets caught up in the cracks and the crevices, it accumulates at the base of reefs, and we essentially think that substrate complexity helps stabilize resource availability for sea urchins. Now, I'd love to test this. I'd love to go out into an ecosystem and manipulate rates of drift retention and availability and removal. And that's just beyond the scope of what we can logistically do. But we can model it. And that's exactly what we did. We modeled the interactions between a grazer, purple urchins, consuming two alternative resources, live kelp and drift algae. And we assumed two primary things. We assumed urchins prefer to consume drift, and the urchin behavior is a function of available drift. So we tested if and how those assumptions, in conjunction with the processes of drift production, retention, and loss, affect kelp forest dynamics, such as stable persistence of the forested state versus switching between alternative stable states. So we're basically going to try to model and see if we can reproduce these types of dynamics. All right, so here's a conceptual math-free overview of the model. Kelp is able to recruit, and kelp dies, and some proportion of the kelp that dies is retained as drift. So you can almost think about this as a substrate complexity parameter. And then some of that drift is eventually lost. It decays or is removed. Urchins consume both kelp and drift. And the behavior, the proportion of urchins grazing upon kelp, I'll talk about the function that governs that behavior. Urchins recruit and are removed from the system. So I'm going to motivate the biology underpinning two functions. The first is the function for kelp recruitment. Don't worry about the math. What I want you to note is that when there's a lot of kelp available, the current availability of kelp is high, there's very, very few kelp recruits. So think about a heavy canopy. There's a ton of kelp, and it shades out the benthos. It shades the seafloor and competitively inhibits kelp growth. And that's why we think we see kelp population cycles. Now, on the other hand, when there's very little kelp present, there's a lot of recruits. So you can think about there's no surface canopy absorbing the light and inhibiting the growth of youngsters. So you see lots of recruits. And I motivate this to say that this kelp recruitment dynamics function is what allows us to see stable limit cycles in the subsequent figures and in the dynamics of this model. All right, second function, the proportion of urchins grazing kelp. So you can think about this as urchin behavior. And similarly as before, now we have drift on the x-axis. When there's very little drift available, all the urchins are grazing upon kelp, right? So the proportion of urchins grazing upon kelp is 1. When there's lots of drift, all the urchins are grazing upon drift. The proportion of urchins grazing kelp is 0, right? So this function encapsulates our two assumptions that we're making about urchin behavior in response to drift availability. All right, I'm not going to go over this too much. I do want to just motivate the system. So we have rates of change equations for kelp, drift, and urchins. And our primary components here, kelp recruits. Kelp is lost or recruits there. Kelp is lost to urchins, and kelp is lost to other sources. Likewise for drift, drift recruits, is lost to urchins, and then is removed. And then urchins simply recruit and are removed from the system as well. All right, I'm going to build up some of the dynamics that we observed in the system. So here on the x-axis, we see time. And so now we're simply solving that system of equations that I just showed, and we're tracking how the state variables, how kelp, drift, and urchins change through time. So we have two steady states or two attractors in our system. There's a low kelp state where there's very little kelp. Oftentimes, this attractor exhibits a fixed point, which is what you see here. And we have a high state, a different attractor that oftentimes exhibits stable limit cycles, which you see here. Now, this model is analytically intractable because of the temporal lag parameter, so I had to analyze it numerically. So think about we solve the system out given a single parameter value, and then we record what is the value of these variables at steady state. That is when the system is no longer changing. So we record that, we plot it here, and then we slightly alter the parameter values of a single parameter in this case, and then we run the system out again and then once more record the steady state values. So we do that across an entire range, and by we, I mean a computer, obviously, across a range of parameter values. And we see here the fixed point, the low state, bifurcates to starting to exhibit stable limit cycles. So that's similar to the low state just suddenly starts to exhibit stable limit cycles at a certain parameter value. Now something different is depicted here. So now we are altering a parameter value, but it exhibits a switch between attractors. So the system at low rates of drift loss, so there's lots of drift in the system, the kelp is exhibiting the stable limit cycles at the high state. Then once drift loss increases, the system shifts down to the low state, and it's also a shift between stable limit cycles to a fixed point. So this is again similar to what I motivated before. This is environmental conditions of the system changing. Now we also see shifts between the two attractors when the initial conditions of the system change. So now I'm going to supply the system with a pulse of drift. So I just alter the initial, the state of the system essentially. And we see that for this range of parameter values shaded in gray, altering the initial conditions of the system switches the system from a fixed point to exhibiting stable limit cycles. So if we provide a massive pulse of drift, even though there's fairly high rates of drift loss, for parts of parameter space, we can recover the high state. So this is the definition of alternative stable states, and our model exhibits them across multiple parameters. Okay, so now we are going to expand this single parameter approach and actually now investigate 2 parameters. So we're going to slightly alter both parameters across their range of values. So there's 10,000 points here, and each point represents a slightly different parameter value combination. So now color is going to correspond to the equilibrium value of Kelp, and color with a black circle as depicted here, that means the equilibrium value is whatever the color is, and the system is exhibiting stable limit cycles. So what I want to emphasize here as I show you some additional, as I layer on additional complexity, is how urchin grazing is key, and the switch in urchin grazing is key to the differences in the stability regions we'll examine. So first off, this area, this is the low kelp state. It's a fixed point. All urchins are grazing kelp. Kind of its opposite, this is the high state. So we have high equilibrium values of kelp. All urchins are grazing drift. Again, a fixed point. And then lastly, once more, all urchins are grazing drift. But now, for this region of parameter space, kelp are exhibiting their recruitment dynamics, and we see stable limit cycles. So now we're going to examine 3 more regions in this gray area. And this is the bordering between the low and the high state. And as we'll see, this is where the system exhibits alternative stable states. Specifically, this lower area is where urchins graze kelp and drift. So they're actually switching back and forth, and kelp are exhibiting recruitment dynamics, and thus we see cycles counterintuitively at a low equilibrium value. And then when the system shifts to the alternative stable state, all urchins graze upon drift, and it's a fixed point. Kelp no longer cycle. This middle area is a little more intuitive. It's simply urchins shift from consuming kelp to drift, a fixed point to a fixed point. And then lastly, this upper area, urchins shift from consuming kelp to, again, as previously, switching back and forth between kelp and drift, and kelp exhibit recruitment dynamics, which is, as you may note, the inverse or reverse switch in qualitative behavior. So again, don't get lost in the details here. The main message is that the behavior, the grazing behavior of urchins is required not only for alternative stable states, but the entire existence of this high state is predicated on the ability for urchins to switch their behavior from drift to live kelp and vice versa. All right, there's a lot of different ways in which we can view the system. There's a huge amount of complexity here, and one can kind of get lost in the bizarre structures that we're able to see. We're not going to go into that. And instead, I'm going to close this chapter with one final example. We're going to examine kelp loss, i.e. drift creation, as well as the retention rate of drift. And we have 3 regions that we just went over. We have the low state, where urchins graze kelp. We have the high state, where urchins graze drift. And we have the region of alternative stable states, where urchins switch from kelp to drift. What I want to emphasize here, the biological intuition here, even when the rate of drift production is as high as it can possibly be, if the rate of drift loss is fairly high or the retention rate is low, there's no amount of drift that I can supply to this system to switch urchin behavior. I can't elicit alternative stable states here, right? So this is similar to what we think substrate complexity is doing in the system. It is stabilizing the retention of drift algae and thereby inhibiting the switch from drift to kelp and thus allowing the forested state. All right, so to wrap up this chapter, we have switching between resources that enables the existence of alternative stable states, and I emphasize the importance of the relative rates of drift production and drift retention. Now, all of these results are based around the assumption that I motivated previously, that urchin behavior is a function of drift availability and that urchins prefer drift. Is that true? Is that biologically supported? Well, that's exactly what we tested next in the final chapter that I'll talk about. So I tested whether urchins prefer to consume drift and whether the density of drift controls that switching behavior. And we formally evaluated whether or not urchins exhibit a switch between alternative resources. So we tested this by deploying these cages and placing varying densities of both of these resources into the cages along with 20 purple urchins to see how those varying densities affected the rates of consumption by urchins. Now, quick shout out to Mark Carr at UC Santa Cruz in the RC lab and Casey Sheridan. We teamed up to conduct this experiment. We ran multiple experiments over a 2-year period. It was a wild time and just an absolute pleasure working with these guys. All right, so treatments. So we had drift algae. My drift were simply blades of giant kelp that we clipped and placed in the cages. And our kelp were actual giant kelp plants that we affixed atop paving stones. Now, the key thing here is we don't think that there's much of a compositional difference between those resources. That is, there isn't much of a nutrient profile difference between our two resources, which are obviously from the same species. What differed is their condition and their context. So the drift sifted about on the bottom of the cage, and we arranged these paving stones such that there would be cryptic places for urchins to tuck into and hide if they so chose. The drift naturally accumulated there, whereas the kelp we affixed atop these paving stones. So this kind of mimicked a rocky reef. The kelp's up on the reef. So for the urchins to consume the kelp, they had to actively wander or move up onto the paving stones to consume it, which, as you can see, they were more than happy to do in a treatment where there is no drift for them to consume. And that is what is depicted here. There's lots of drift in there, and they're going to town. All right, so treatment structure. So I had 8 different levels, so 8 different densities of drift algae, and all of those cages had a single macrocystis plant, and the squares are cages, I should mention. And then I had 8 more cages with the same densities of drift, but now with 4 plants within. So again, we're trying to tease apart the relative effects, excuse me, of the densities of these two resources upon the rates of consumption. All right, treatment structure across time. So we go out there, we weigh all these resources, we place them in the cages, and we let it run. And urchins do their thing. We come back 48 hours later, we pull everything out, we measure it, and we count or we record how much biomass has been lost in this period. And then we restock the resources back up to their original levels. And then we put the resources back in and let her run once more. We do this entire process again. We pull all the resources out, we measure biomass lost, and then once more restock the resources back up to their original densities and let it run. And then finally, we pull everything out, measure it, and do not restock. So we restock the resources up to their treatment levels 3 times, and we have 3 observations of kelp and drift loss. Now, we ran this experiment 4 times. We got new urchins for each experimental run, and we shuffled treatments across the cage array to counter any cage effects. And so these 4 treatments we can lump together. These are essentially independent replicates that we can use to visualize and analyze the data. All right, we fit a model to these data. I'm not going to go over it in too much detail, but this equation represents drift remaining through time. So you see the negative sign, drift declines as urchins consume it. Kelp remaining through time, again, negative sign, kelp declines as urchins consume it. And then gut fullness through time. So this is something we didn't actually measure. Instead, we modeled how consumed resources would fill an urchin gut. And the reason we did this is because we pulled urchins from an urchin barren. So these urchins were ravenous, they were starving, and we might expect that a starving urchin would exhibit really, really high consumptions initially, but they might start to get full. They might kind of slow down as they no longer perceive themselves to be in such a starving state. So this gut fullness through time allowed us to model and scale the consumption of drift and kelp. So as this gut fills up, consumption declines. And last thing I want to motivate here, this expression here, this logistic expression contains parameter Q. And so that's a resource switching parameter. So if urchins exhibit one resource or another, we'll be able to quantify the direction and magnitude of that with our estimate of Q. Okay, so we did this experiment at Hopkins Marine Station, Monterey, California. It was absolutely beautiful and perfect for this type of experiment. 15 feet of water, sandy bottom, sheltered from large wave events. Casey and I were doing 2.5-hour dives on high pressure 120 tanks. It was just a wild time. Sometimes it looked like this. Some days it was just absolutely stunning. But it was a blast the entire time. Now, I'll be honest, folks, the first summer was a complete bust in the sense that our cages, ironically enough, were inundated with drift from outside. So you can see grasses and drift algae here. It just completely messed up our treatment densities within the cages and threw off urchin behavior. So year 2, we completely encased our cages in fiberglass window screening. So we essentially had a double hull of netting and then this outside layer of window screening around the entirety of the cage. And it worked beautifully. It prevented the entrance or removal of anything the size of a mosquito. I mean, this is window screening, but it worked really well. Water movement was still able to get through. So we were able to hold our densities precisely at the desired biomass initially. All right, let's jump into some data here. So the 3 columns are the 3 temporal periods, that 3-period sequence. And so we have drift available and then drift consumed. And as the availability of drift increases, drift consumption increases. And then you see this temporal decline. So this is our hypothesized. This is starving urchins kind of starting to fill up and no longer perceiving themselves to be quite as ravenous. Let's look at kelp consumption once more with drift availability on the x-axis. As drift availability increases, kelp loss declines. So kelp is primarily consumed when there's no drift, and virtually ignored, kelp is ignored at high drift. So okay, let's look at kelp consumption relative to kelp availability, and we see nothing. There really isn't a pattern here. Kelp consumption, according to this experiment, is independent of kelp availability and instead is controlled by the availability of drift algae. All right, what about our preference? What about switching behavior? Well, we can plot our data and view that. So we can view the proportion of drift consumed relative to the proportion of drift available. And as we see, drift is indeed preferentially consumed. We don't see any switching across this line, so there's no positive or negative switching. However, we do see evidence for rank switching. So that's a switch in the proportional consumption of a resource relative to the total biomass available. So relative to drift plus kelp availability. So we see kelp is preferentially consumed at very, very low total biomass levels, and it switches to drift at higher levels. Now we'll strip away kelp availability from this figure, and let's just look at the availability of drift. And we see that around 50 grams of biomass is where urchins switch from kelp to drift. So this is we formally captured in an experimental sense a preference to consume drift and the point at which urchins switch their behavior from kelp to drift. Okay. So I'll just briefly mention we did fit this model in STAN and we estimated posterior distributions. I'm not going to go over them in depth. I will just motivate that our resource preference parameter was estimated at a little over 20. So this is a massive preference to consume drift. And that's not a surprise given the observational data we just looked at. And so what I did next was then simulate using these posteriors. Let's simulate this system and see if we can recreate, so to speak, the data that we observed. So how well does this system of differential equations describe the observational data? And so here we're now plotting simulated data using these posterior distributions. And as you can see, for drift loss and kelp loss, it's a pretty decent approximation of what we actually observed. So the parameter values that we estimated using this model can simulate and reproduce these patterns. Now, there is no empirical equivalent for stomach fullness. Again, we did not measure this. This is something we simply use to model the manner in which a stomach, it's not an actual stomach, but the gut contents of an urchin fill up and accordingly decrease consumption across the 3-period sequence. And ask me about this quirky little dip in stomach fullness if you want. It's an expression of suboptimal foraging behavior, essentially. Urchins prefer drift so much that they switch to that and consume that even at the expense of more kelp being availability. So basically they take a loss in net consumption in order to abide by their strong preference. And then likewise, for the preference and switching behaviors, our simulated data align well with what was observed. Here I simulated with 300 kelp and 300 drift initial conditions, 300 grams. And so you can see it aligns. It's a bit off there because there's so much kelp available, but then when we strip away the kelp, we get pretty much the exact same switch point in consumption when it's just drift plotted. Okay. Folks, we're getting through this. So concluding this chapter, we tested the key assumption from chapter 3 that urchins prefer to consume drift and that the density of drift controls the behavior of kelp. Urchins indeed exhibit a switch, and we were able to fit a model and estimate parameters for switching, and we're able to model approximately the temporal decline in consumption across the 3-period sequence. So returning to this and just starting to conclude, we talked through how these low complexity locations exhibit these shifts in state, persistent shifts across 10 years or so. And I'll just fill this in a little bit more. We think that these low complexity locations are just subject to losing their stabilizing feedback processes, such as the processes of drift creation, retention, and loss, just through perturbation events. The entire kelp creation or kelp growth and drift creation is just removed. And then urchins are able to stabilize that state, whereas similarly perturbed high complexity locations do not exhibit those switches. I didn't mention it before, but predators also do associate with substrate complexity. So they're more abundant in highly complex locations and less abundant in low complexity locations. And this adds an additional, kind of strengthens the ability for urchins to exhibit their switch in behavior and for that switch to become persistent, leading to persistence of the urchin-barren state. And then I also didn't mention previously, but once that state switch occurs, there are additional feedbacks besides urchin grazing which stabilize that state. So urchins keep the benthos clear of kelp, encrusting algae spreads. Encrusting algae is known to produce a chemical cue, eliciting urchin recruitment. So this just strengthens, so more urchins are entering into the system essentially, which further stabilizes the barren state. I think there's future directions to go with this research. How widespread is this pattern? I don't know. It could be specific to San Nic. It could be applicable more broadly. I simply do not know. But I think it's worth looking into. And then concluding chapter 3, so I talked about how switching between resources enables the existence of alternative stable states and these very distinct dynamics that were exhibited by the model, and that the rates of resource creation and retention are important to understanding system stability. And that's our hypothesized substrate complexity parameter. I think there is more ways in which we can expand upon this model. And if you want to talk to me about that, I'd be more than happy to answer questions. And then lastly, concluding chapter 4, I'm excited about these experimental results. They are dramatic. They were unexpected. Obviously, the inference is restricted to a 1 meter cage, right? We don't know how applicable this would be if we went out into a kelp forest and tried to maintain a pulse of drift, how that would affect urchin behavior. I think that's a logical next step. Experimental removals of natural reef, experimental removals and placements of drift to see how that affects community structure and urchin behavior over time. And with that, folks, thank you very much for listening to all of that. I want to thank my committee. Mark, it has been an absolute pleasure working with you. I have learned so much. You have changed and helped shape the way I think about the world. And yeah, I can't thank you enough. It's been an absolute pleasure. Tim, you opened up worlds of opportunity for me back in 2012, I think it was, when we started working together, and I really appreciate it. Bruce, thank you for your feedback on my dissertation. I am just in awe of what you have put together over the decades, and I really appreciate your support on my committee. Julia, your geospatial class changed the way I think about pattern, and just I really enjoyed your classes. Some of my favorite classes at Oregon State for sure. Ginny, you were kind enough to swap onto my committee in a time of need when someone else had to switch off, and that really was a lifesaver. So thank you so much. Jim, you have been my GCR. Thank you. I know that the subject matter is likely quite different from what you're used to, but I appreciate your time and support as my GCR. I want to acknowledge my funding sources. The Dr. Earl H. Myers and Ethel Myers Oceanographic and Marine Biology Trust. I want to acknowledge NSF, National Science Foundation, PSCO, Oregon State, and the University of California, Santa Cruz. And also one more shout out for Embroideology, SB, considering I used her design all over this talk. She was very kind and excited about allowing that. So thank you. I also want to thank USGS and the entire California sea otter world and the broader sea otter world. You are all not represented with these photos. This is mostly a USGS Sand photo, but this is a shout out to that world. Mike, you brought me out to Sand I think in 2011, 10 years ago, and that fundamentally shaped how I thought about ecology and deciding to pursue grad school, and you supported that, and I really appreciate your friendship and kindness and support over the years. Joe Tomaleone, we have had a blast working in the field, joking. I've really enjoyed my time working with you, and it's a treat to see you and Nicole establishing your lives and flourishing now. And shout out as well to Ken Collins and Sean Larson in here as well. I've really enjoyed our conversations and time working together, and I'm really hoping we all are able to get out to San Nick 1 month from now. We shall see. All right. Big thanks to Mark Carr and the Ramundi Carr Lab at UC Santa Cruz, and Casey Sheridan for supporting this work, and Casey, my partner in crime, teaming up with him. We couldn't have pulled off an experiment like this without a lot of really long days. So thank you, Mark, for supporting that. Thank you, Casey, for teaming up with me, and then all our volunteers, especially Jessica Bray. She dove with us both summers, and I'm not sure we would have been able to pull it off without her support. And likewise, shout outs to Anya, Haley, Hannah, Benji, Samuel, and Bryant. And I want to acknowledge as well the IB community and the friends and the good times along the way, especially pre-COVID, before I went into this little box that you now find me in. I really enjoyed working and joking and hanging out with all of you. Shannon, our office started with humble beginnings, and it turned into an epic plant explosion, arguably out of control. But it was glorious, and it was a wonderful place to work, and I really enjoyed our conversations and talking with you and sharing an office with you over the years. Rachel. So Rachel and I started dating in November of 2019. And as some of you may recall, in the early parts of 2020, our world changed in certain ways. And I'm not sure how I would have gotten through that in my little box were it not for the adventures and hanging out and time together with Rachel. So it has been a pleasure and a heck of a time to build a relationship. But thank you. And thank you for putting up with me over a apparently 2-year period at which I decided to no longer get haircuts. I'm not sure what happened, but thanks for sticking through it. And then lastly, I want to acknowledge my parents. They have been there from day one, and they have supported me and loved me and encouraged me to dive at an early age, and that opened up pathways. I really appreciate their support over the years, folks. You really helped make it happen, so thank you very much. And with that, with any time remaining, I will happily take any questions. Thank you all again for tuning in.
Well done, Zach. Very nice talk. Yeah, I will try pull people's questions. I believe you should be able to either raise your hand or type in your question into the Q&A box, into the chat, or not chat, but it should be a Q&A box. And I'll call on people. We're pretty much out of time, but I think we should squeeze in a couple of questions if this is working. Seeing comments coming in through the chat. Let me know if you have questions.
Thanks, folks.
I see Francis. Let's see if I can see your hand raised. There. Talking permitted. Okay, got it. You should be able to go, Francis.
Okay. Hey, Zach. Beautiful work. Beautiful work. Congrats, man. I want to just ask you a little bit about this. The very formal notion of alternative stable states is kind of it's same environment, same kind of abiotic factors, but kind of divergent biological communities. Does it matter, conceptually that it's what's driving your system is really this external input of drift? I'm assuming that it's not super closed system that the kelp dies and immediately becomes drift right there. So does that change your conceptualization at all?
Yeah, that's a great question. A couple of things I want to touch on. Just the conceptualization of alternative stable states is incomplete in the sense that we know the environmental conditions are changing, right? So just that strict notion of only the community is changing, and that's what exhibits alternative states. That's especially now with climate change and the increased frequency and intensity of disturbance events. In terms of the input of drift, I think that is going to absolutely affect what we see, different rates of input. And that's where thinking about substrate complexity, modifying the rate of drift that is retained, including that which is externally delivered from elsewhere. And it might also be that it's not always substrate complexity that's doing that. I think there's examples of locations where just the configuration of the oceanographic processes and the local shoreline may have relatively consistent deliveries of drift algae, and it may be completely flat, but drift just happens to accumulate there. And that's essentially the same process that we're attempting to describe. That's what substrate complexity, we think, does at San Nic. So I'm not sure if that answered your question, but that's kind of my immediate response.
Yeah, no, thank you. Yeah, no, that's it is a very formal, strict definition that we start from.
There's a question in the Q&A or in the chat box. I was wondering, when determining the drift preference of urchins, that is your experiment, I assume, were there any predators present that may have affected the consumption rate of attached kelp?
Yeah, great question. I should have mentioned that. There were no predators in the cages. So in terms of actual predators being present within the cages, the answer is no. Could there have been predators lurking outside? Could chemical cues of a predator have been present? Absolutely possible. Nothing we can do about that, except put the entire experimental array in a sandy area, that's not a rocky reef and hope that there are going to be fewer predators in that sandy spot. So the attempt was to strip away the influence of predators or any other species. So it's just the urchins and the varying densities of those two resources that determined, as best as we could estimate, the consumption rates.
Noah and then Emily have questions. Noah, go ahead.
Hey, great talk. Great, combination of theory and experimental to back that up. So you talked about how, the gold standard experiment, the one you'd want to do if you had all the money and resources in the world, is to experimentally kind of do this. Could you talk a little bit more about, or to experimentally like alter the landscape and see how that affects it? Could you talk a little bit more about that? Maybe, do you have any intuition about like how long it would take you to get results from something like that? And just kind of like flesh that out a little bit more.
Yeah, thanks. No, that's a great question. Yeah, there's a couple different ways that I think one could go about it. So you could try to manipulate. Oh, okay. I'm not sure. I don't think I'm muted, so perhaps people can hear me. Okay. Well, there's a couple different ways that I think you could go about it. You could manipulate substrate complexity, so you could try to build artificial reefs and see if that captures drift and kind of captures some of those feedback processes. Or you could test the effects of drift on existing locations. So you could go to high relief areas, low relief areas, and place a bunch of drift at those locations and try to keep it there, sustain it, see if we can reproduce the effects of substrate complexity even in a low relief area. That would be ideal, to go to these flat locations, see if we can reproduce the effects of substrate complexity. That kind of strips away the substrate complexity component and gets right to the mechanism that we're envisioning, the altered rates of retention. So I think sustained additions and removals of drift would be really interesting to see, and it'd be interesting to see how urchin behavior responds over the long term. And such experiments might also help us disentangle what other processes are at play that are driving these effects. It's surely more than just drift retention. There's likely competitive effects as well. There's more species present on these high relief structures that may influence rates of urchin larva survival, for example. And so it'd be useful to try to evaluate some of those other altered processes that associate with substrate complexity.
Emily, you want to squeeze in a quick question? And then we should probably wrap it up.
Okay, can you hear me?
Yes.
Okay. Yeah, so quick question. I'm sorry, I was a little late, so I didn't get your whole discussion of the substrate complexity and how it was measured. But I was just curious about whether you were able to correlate mapped substrate complexity, like from the multi-beam, with what you actually measured in the field.
So no, I did not correlate. That's a great question. And that's a future direction we want to go with the research. There are side scan sonar data. So I'm just scrolling. Okay, here's how I measured it with this little gizmo device. There are side scan sonar data for sandnik, but they're at the 2-meter grain. So there's basically 5 data points per these transects. And I just wanted to get a little more information, high resolution information about the substrate complexity. Hence I use this little wheel device. And that establishes the basic relationship between these precise slices of seafloor and the associated kelp forest dynamics. The next question, how broad is this pattern? Do we see this elsewhere? What do we see at larger scales? That's when absolutely the bathymetry, the underlying acoustic imagery, could be hugely informative. And that's definitely a future direction that I'm excited about.
Oh, and great job, by the way. This is really cool.
Agreed.
Thank you.
Thank you, everybody, for joining us. Committee, let's take a few minutes break before reconvening. Teresa kindly sent us a new Zoom link so that we can all see each other's faces. So sign off of this one and join that new Zoom link that should be in your inbox. Let's reconvene at 1:15. And yeah, well done, Zach. Thank you, everybody, for joining.
Yes, thank you, everyone, for joining. I appreciate your time.